So I'm really thank you. Thank you for inviting me here, and I'm very happy to share with you today a new research, relatively new, but I think very important and incredibly very important research area which changing our biology, the view of our biology. It's a microbiome research. And correct, I will talk about the Estonian cohort data today, but first part, I would like to also give some more introduction into this topic that I think not everyone here probably working with this field. So the first part of my talk will be more general and introduction, and then I will share some of our works which we have done in Estonia, in University of Tartu, using Estonian Biobank data. But first of all, I wanted that you will think about 10, 15 years ago when you heard words like bacteria and viruses. Probably most of you were thinking something happened with my health and I had to get rid of those organisms. And you were probably quite surprised hearing that all our body surfaces are populating this huge amount of millions of different microorganisms, which we call a microbiome. And those organisms can be bacteria, archaea, fungi, and viruses. And there is a long history to study bacteria, and it has been changed tremendously during the last 20 years ago when we start to investigate those microorganisms using a DNA and sequencing, using the same technology we use for studying a human genome, using next generation sequencing and analyzing now a DNA or metabolites or RNA level and the proteomics level. So there is a huge steps and exponential data which came out because of these technological improvements which we had last 20 years in this field. So we have done some comparisons now to see what we have here comparing our genome and the microbiome genomes. And if we count the number of cells, then we have almost equal number of cells compared to human cells and the number of microbial cells we have in our body, right? But when we look at genes, gene content, there is a huge difference, right? So every human gene, there are approximately 100 times more bacterial genes, or not only bacteria, but all those other microorganism genes, which gives a really huge potential, functional potential for our body to interact with our body and have an influence to our health and behavior. Doing more those comparisons, when we look our genes, right, and compare them, we are very close, 99 and even more than 99% similarity. But when we type the microbiome, it's vice versa. We can be only 5% similar, which means that we have all very, very unique microbiome profile. And this can change also because of several factors affecting actually microbiome community. Diet is the first, then our age, our environment and medications, our health, everything can change all the time and constantly our microbiome community. And this makes a huge difference when we study the microbiome. So that the first data sets which really gave us the more view how many microbes we have and how many different microbes we have in our body was the two big projects which was established almost the same time. One was a human microbiome project, NIH-funded project, and another one was the same time in Europe, MetaHit projects. And those were very important projects in that time because this was the first big sequencing projects. And if we look at now the numbers of those individuals, those projects they did, those numbers are really low, right? We cannot publish now probably the population-based data using these low numbers. But those were the first studies and gave us really important knowledge. And this ESMB project showed them they investigated only 240 individuals, but they collected microbiomes from different samples from different body sites. And it illustrates very clearly in this study that each body site has a very different microbiome community, which is logical because there are different niche for a microbiome. And the other study, which was a meta-hit study, actually this NIH study, they used that time only sequencing very specific gene, which is only for prokaryotes, and a very small part of this gene actually. So this was quite low level of the data which they had, but very important first picture about the microbiomes. So the meta-hit study, they used the whole genome sequencing not the entire bacterial genes, also the lower level, but not only particular one gene, but covering most of the genes in a microbiome. And they used even less samples, but they used some disease samples and healthy control samples. And this study was the first study looking this functional profile as well, not only taxonomic, because this 16S-based sequencing, the most information you get is who is there, what are the species in human body. But here they could see also the functional potential and the first time calculating the numbers that how many genes we have encoded by bacteria, and showing that some of the genes like carbohydrate metabolism and amino acid metabolism are more coming from the bacteria side. So we're born in a sterile case, but how we get the microbes is we first get the microbes from our mother after birth, and in the beginning of our life, the microbes, we have very low microbiome diversity. Only certain very specific bacteria the babies have, which they need for digest the milk. But this diversity starts to increase, and by the age actually up to 3, we have more or less the adult-like microbiome community already in the gut. And during the lifestyle, many factors can affect, which I already mentioned, microbiome diversity, and then diversity again a little bit decays in the end of in the elderly. So it's very important, the studies have been really shown right now that it's very important the window of the first 3 years of life. And why? Because the microbiome helps to develop immune system. And that's why you have heard probably that the babies who are more raised in a natural environment, they have more diverse microbiome, which helps them to develop a proper way the immune system. So now if we look just looking in different perspectives, when we look one individual, we know that in the gut we have almost like approximately 500 different species of bacteria. I mostly talk about today in a bacteria. This is mostly covered area, and we actually in our research also working in a bacterial side right now. And as I mentioned already, we have a huge number of genes, and also they produce many, many metabolites in our body. But if we think about in a population level, then we can see that those numbers accelerating, right? It's a huge potential to really think about what those genes and the gene products and metabolites will really do in our body. So we have different types of habitants. We have some very dominant ones, which most of us probably have. And then we have some rare, not some, but many rare species. And we have also sometimes in a life transient species. So there is a whole ecosystem which is really living together and producing a huge number of functionally important molecules. So now why do we need a microbiome and gut microbiome? There are, I would say that two very important functions. One, of course, is immune system defense, which is really crucial for early development. And also it stimulates the whole life, the immune system, and also it gives a protective against the pathogens. But we also know it's the microbiome in the gut. It's important for our metabolism. Microbes, they produce a lot of enzymes, which we don't encode in our genome, and which helps to digest certain food. and it's produced a lot of vitamins and hormones and many other molecules which we probably have heard about, short-chain fatty acids and vitamins and the hormones, which I already mentioned. So it's the whole system which is interacting to each other and helps to digest and function properly our gut and also communicate with other organs. So Hippocrates already told very long time ago that all diseases begin in the gut. And that's, I think, quite right if we think right now that we probably don't find any phenotype if we type in a Google and try to find connection with a microbiome. So microbiome is really affecting all organs of the body. This picture is very tense, but it was published recently in a Cell that just showing that every organ in our body and the system, they have connections already found where the microbiome associate with some different diseases, like of course the metabolic diseases, the first obesity and type 2 diabetes and metabolic syndrome, where those phenotypes which were studied in the beginning, or the gastrointestinal diseases, but also many other ones. And there is a connection with the brain, which you are mostly interested in here in the consortium, and more works coming with this area as well. So there is also hypothesis and idea behind that the chronic diseases we have right now spread, like type 2 diabetes and many cancers, they have been linked to changes in our lifestyle and the microbiome. But most of those, the data which we have our hands so far are mostly associations or correlations, and only recently more works about the functionality and mechanisms start to come aboard. So it's really important to now move more from those associations to see really the effects of the microbiome, and I believe those works will start to become available very soon, and already we can read some very nice works from recent literature. So here I also had, before I moved to my work, to some gut-brain connection and mental health, because in this consortia you probably are more interested about that. So we know that gut microbes influence neurodevelopment or modulate behavior or contribute to neuropsychiatric disorders such as schizophrenia, autism, depression, Parkinson's disease, Alzheimer's disease, and so on. So there is idea behind that there is a bidirectional linkage between a brain to gut microbiome, and those can be like the signaling can be through the nervous system, through the metabolites gut bacteria produce, and through the immune system. And we can also see here in this graph that there is an increasing number of publications come in this area as well. So many studies really, I think I show the next slides, show microbial derived molecules that signal brain via neuronal pathways of the vagus nerve or modulate through the immune system. And there are also hypotheses that behind that, since the gut is really a central part of the brain diseases as well, then there is a huge potential for novel therapeutics for central nervous system disorders as well, because we can reach more to the gut to treat those brain diseases. Then we have to go to the brain directly. For example, Parkinson's disease, which is one good example for treatment problems. So this is actually the recent review from Sarkis Masmani and colleagues group in Nature Reviews, showing that there are microbial derived molecules that modulate host behavior and nervous system functions. And here, this figure really represents some examples, and some bacteria, we really know that they will produce important neurotransmitters like GABA and also other ones, and some connections with mental diseases and disorders. And there, like I already mentioned, there are several ways to do that. That there is, like, for example, this lactobacillus rhamnosus producing gamma-aminobutyric acid and also changing this gamma-butyric acid receptors. And through this system, there is a less stress and anxiety and depressive symptoms like behavior. But most of the studies, as far it's not my area of the research, but what I really understand that most of those studies in the mental health and neurodegenerative disorders, they come from the animal models still. So the data from the human population is still lacking a lot and need to be improved. But there are like many studies from animal models showing how when we cut the vagus nerve, how we can get rid of those microbiome-derived molecules pathways. The next slide, I wanted to say that many studies has been done in a mouse model, but there is a lacking data in human studies. But some like autism syndrome diseases, there are more works than other phenotypes available already in human studies. And if we think about now in human microbiome studies and where we are in those studies, which I introduced at the first sequencing large scale, in that time sequencing studies was published in around 15 years ago, then now we see that the number of these publications has been really increasing, like 5,000 to 7,000 in a year right now, studying the microbiome. Then we have much more knowledge about how the microbes is related with different diseases or drugs affect microbiome population. Also, we have some data already how the cancer therapy, for example, respond to your microbiome community. But we can also say that the microbiome field is still very exploratory phase, and we really need still to develop standards and how to analyze this data. Like it was telling in the introduction that there are a lot of hype to microbiome as well, and this comes because in the first 10 years of the microbiome field, we didn't know how to really analyze this data, how to collect this data, because a lot of things really affecting the results. And now a lot of efforts, I would say, the last 2-3 years have been putting in this area and standardizing more how we should work with the microbiome field to analyze this in a proper way so that we can really replicate the studies between different populations. And also in the first studies, we had very broad view of the microbiome in that sense that the studies were done in analyzing a very specific one target gene, which was 16S RNA sequencing. But now we go more deeper to do deep whole genome sequencing, which allow us to go more in a strain level analysis to know what exactly the very specific strains are, and also going to variation studies, like all the bacterial genes, they have also the same, like in our human genes, single nucleotide variants, copy number variants, so we can start to go really the similar way to study the microbiome as we study in our genome. And we also need to focus more, not only bacteria, but also the viruses and fungi and archaea, and this work's also coming out now. And a very big field of the microbiome is now the pharmacomicrobiomics, how the microbes can change the drug metabolism and affect that. And I will come to these studies later. But there is a really thinking about going more to therapies and applications, but I think it needs more data to be able to go towards those aims right now. If we do the microbiome study today and we collect samples today, so what it will tell us, right? We know that the microbiome has been affected, which I already mentioned, for several factors. From past, it's like I said, like in medication uses or dietary or surrounding environment or lifestyle and so on and so on. And this is all reflecting our today's microbiome. And if we go further and we have the microbiome samples today, what we can tell your health for tomorrow? That's an important question, and that's something which we're really interested in starting here, to be able to say, based on your microbiome today, can we predict what kind of medications you are able to metabolize and it's good for your body? What kind of, can we predict some diseases based on your microbiome data? And also there are studies also predicting what diet or nutrition is best for your body based on your microbiome. So those are really important and questions we ask and we need to investigate. So now I'm moving to the studies which I have been done in Tartu. And then starting with these general slides introducing Estonian Biobank. It's a population-based biobank established already 20 years ago in Estonia, and currently it has more than 200,000 individuals, which is one of the biggest population biobanks in terms of population size. It's about 20% of population, covering Estonian population. Establishing this biobank, the first was very important that we had a good law. We established a good law, which is Estonian Human Gene Research Act, which is actually provide us as a scientist a very good hand to do research, because all the people, it's a voluntary-based biobank. All the adults, like more than 18 years old, people can join with this biobank. They give a very broad informed consent. So that means that we can always recontact those individuals, collect additional data from those individuals, and we also have access to electronic health records. It's a very nice study, but when I came back to Estonia and started to work with a microbiome during my postdoc stage, we realized that we're missing a microbiome, right? We cannot tell everything when we only have our genome, because, like I said, microbiome is a super important part, and we need to consider this as well. So we initiated the Estonian Microbiome Cohort in 2017. It took like almost 2 years when we collected the samples, and we were able to collect more than 2,500 individuals, and which covered the whole part of Estonia, which is also, I would say, one of the rare population cohort covering the whole country, not the very specific areas of the populations, which is quite common in the bigger countries. And all those participants, they donated fecal samples, oral samples, and plasma samples. Today I will present the data which is only based on the gut samples, and we sequenced the whole genome sequencing in the gut samples. And what we additionally collected, we collected also self-reported questionnaires, and all those individuals actually joined with a biobank already 10 years ago, when they filled the self-reported questionnaire, and we asked them the information about the health, medication status, and also the lifestyle, dietary, physical activity, and so on. So they reported the same questions then 10 years later when they joined with this microbiome cohort. We have access to electronic health records, and which means that we have access to the medical history, every procedure those people have had, different medications. We have a hospital records, and also access to different registers. So if the country has some screening programs or cancer or death registers, we have access of those. So this is actually showing you now just example. If we have one person and we collected a microbiome sample in 2017, we can see the past, what is the health profile of this person. It's just arbitrary profile to show that those ICD codes, what diagnosis this person have had and when, and also what medication this person has been taking past. But what is also important, that we can also see the future. We can see now, after 5 years collecting a microbiome sample, what have happened now within the 5 years period after donating these samples. Now a little bit the results of this study, because the study was published recently, and most of the analysis was done in very talented researcher, Oliver Aasmets. And first I will this is very brief, first question with what we had was very general. What looks like Estonian microbiome, gut microbiome? Do we are different compared to other population or similar? And of course, we were very similar, right? So we are Western country as well, eating the same food, living more or less the same environment. And what you can see here, this data was already known for the first population studies, that if we have a subject, so 2,500 subjects here, and we have a relative abundance of the bacteria belonging to the phyla, then we can see that in a gut we have two major phyla, like Firmicutes and Bacteroides, and 95% of bacteria, different strains, belong to those two phyla, but we see huge variation. And we see that, for example, this person has a 90% or even more those Bacteroides, and this person here and vice versa. But when because we did the metagenomics, we can see the functional profile, the genes. And at this level, it was also shown before that the functional profile is more stable compared to variation. So those bacteria and different bacteria we have, they produce the same functions more or less in this family. So if we study the microbiome, one of the things which you look first is the whole diversity. What is the diversity? More bacteria we have in a gut, the better we have. We heard we need the diversity because diversity has been reduced because of our environment and our health conditions, and all this has been changed over the years. And before I'm going to show the data, I just explain briefly what we mean about diversity. There is two major levels. We measure diversity. First, we ask how many unique species each of us have if we type those different individuals. And then we call this alpha diversity, and we can correlate with diversity index with a phenotype we are interested in. Here is example, like for example, hypertension, and to correlate, and we can see that there are different matrices that it's a negatively correlated with diversity. Then less diversity we have, then if we have a disease. And this is what we always usually see when we study the different diseases, that the diversity is reduced, and we call even the dysbiosis, right? So the second measure we can do in a whole community level, we can count beta diversity, which is how similar the microbiome profiles are between different individuals. And then we can see, we can estimate the variation in between the different phenotypes, how much variation this specific phenotype or trait gives to microbiome variation. So what we did, we used this electronic health records and studied many factors, and also the self-reported questionnaire, and analyzed almost 300 different factors, which goes to the different categories like medical procedures, drugs, diseases, dietary factors, and so on. And we were able to identify more than 100 significantly different associations with the gut microbiome. And here are those measures which I showed the previous slide, that the alpha diversity, which we can see really, it correlates very well with those measures which we call, for example, bad health conditions, less physical activity, eating more sugars and soft foods. We have a lower diversity, the negative correlation with different alpha diversity measures. But we detected many already known associations, like, for example, the biggest variation the gut microbiome gives, the gut empty frequency and BMI, also eating habits or several diseases and drugs. So this was kind of already known for previous studies. For example, one of the landmark study was a lifeline study a couple of years before, showing the same, that there are several factors, all those factors associated with different diversity levels. But here is one very big bar, which is a drug, and maybe you can guess what does it mean? What is the drug which is really affecting the microbiome variation? If someone have a guess? Exactly. So antibiotics. And what we did in this study, we know that antibiotics is really affecting the microbiome, and we wanted to take advantage of this Estonian microbiome data and see not analyzing the antibiotics the same time point we collect the samples, which people usually do. They ask, how many times have you taken antibiotics, and taking this into account when you do the analysis, because we know that antibiotics change microbiome diversity largely. So what we did, actually, we excluded those individuals in this analysis, those individuals who has been taking antibiotics half year before we collected the microbiome samples. But we were looking at these electronic health records 10 years back and wanted to see what is the effect of the long-term or additive effect of uses of antibiotics in the past. And we were quite surprised, actually, that when we do this and clustering individuals, how many times they take antibiotics, and people who already started to have been taking antibiotics 3 or 4 times within the 10 years, we can see that the microbiome diversity significantly already starting to change. And we can see this both in alpha and beta diversity. This was surprising also because when we think about Estonia, it's one of the lowest countries prescribed really antibiotics. I think Netherlands is the first one in that sense. So there is a huge additive effect using antibiotics in the past. And of course, this is just a correlation or associations. And we were really interested to see further what this really mean to our physiology, our gut health. And we set up a study. Very talented PhD student Kertulis was designed a study where we took those individuals, stool samples from those individuals who has been taking many times antibiotics over the last 5 years, and then the groups of healthy individuals who haven't taken antibiotics the 5 years period. And we used the mouse model and did a fecal transplantation, giving the stool samples to the mouse. And then what we did, we measured different mucus functions and the physical properties in the gut. And the study was done in collaboration with Björn Schröder group in Umeå University in Sweden. And this study was not a long study. It was a couple of weeks only. And we really could already show the differences in the mucus layer, which was dysfunctional for those mice who got stool samples from those individuals who has been taking several times antibiotics. We measure the mucus groove and we measure the penetrability. And if we think about the mucus, it's super important parameter in our gut because mucus is a physical barrier that protects against the pathogens and the toxins and penetrability to our bloodstream. So this study, we're still analyzing the data. It's almost done and hopefully publish soon this data. So it was important for us to go further from associations to see really what the functions, but the mechanisms, of course, need to be clarified, and then which we do also analyzing the full data set. So first, I told that we did associations with diversity measures, but of course we also looked up associations with each of the bacteria which we detected with different diseases, and we defined several significant associations, which is shown here in diagnosed, and some of the diseases also which we had significant associations. But what we also see that several diseases had we detected same associations, and it's called also in the literature, it's kind of common dysbiosis between different diseases. For example, here we had a hypertension and anxiety disorder, quite a high number of significant associations, and we can look this also the other way. We did this analysis another way, looking at species co-occurrence network, which means that the bacteria in our gut, they don't live alone there. They communicate with each other. So we created the networks, and we clearly showed the same picture as I already indicated that for example, some diseases like anxiety disorder and essential hypertension, we had the same positive association and same associations between those diseases. And one of the example was a type 2 diabetes, which was really standing up differently. So now if we take this knowledge which we got from this antibiotic study, looking at this additive effect of antibiotics, we actually used the same analysis, but now in this model we took also account with antibiotic treatments, and the picture changed dramatically actually, and you can see that many of those associations really disappearing. And what did this tell us is that that a lot of associations actually we see and we saw here in the first step was confounded by antibiotic treatments. So it's really important to consider when you do the analysis and association studies, not only antibiotics, but also the other drugs. And we also wanted to take advantage and see the prediction power. Can we use this antibiotics knowledge also to predict diseases? And here we use this machine learning models to do a model where we counted only antibiotics usage and the microbiome, and you can see that in some diseases the antibiotics can predict the same, or and sometimes even better than the microbiome. And type 2 diabetes is again exception, where we see that antibiotics information really doesn't account. Why we did this, and it was like interesting for us, is that sometimes we know that for a prediction, from the prediction analysis, it will be really good to have some very good marker, and sometimes maybe we don't really need to analyze the microbiome, but we can get the same analysis, perhaps asking some additional data, for example, how many times we have been taking antibiotics in the past. Gut microbiome and drug interactions are really important and interesting topics, and here I just pick up two studies which I found really landmark studies in that area, which were published in Nature a couple of years ago. And there are two different ways how these interactions can be. There can be that drugs affect microbes, and there is a Lisa Meyer study in MBL, and they tested over 1,000 of different drugs, and they showed that 78% of antibacterials and 24% of human-targeted drugs. So very direct impact on the group of many commensals, which means that a lot of different host-targeted drugs has the same effect like we have in antibiotics. So it's really affecting the group of different bacteria. So there can be also a different direction, that the microbiome is affecting the drug metabolism. And there is a Michael Zimmerman study, showing exactly that they tested more than 200 different oral drugs and different bacterial strains, and could see that 65% of drugs are really metabolized by bacteria. I just gave here one example. For example, Parkinson's disease treatment, levodopa, which is very common, that certain bacteria, if you have in your gut, these gut bacteria enzymes are capable to degrade the drug before it reaches to the brain. And this is just one example, but there are a lot of other examples exactly showing the same. And as I showed in our study, we investigated the additive effect of antibiotics. There was a study recently showing with a Metacardius cohort with a large populations that there is an additive dose-dependent drug effect to many host-targeted drugs as well. And that is the area we really wanted to go further, and analyzing the other drugs data which we have available in Estonian Biobank, and did this microbiome drug associations, looking the information about how many times people in the past have been taking the drugs, and counting this additive effect, and looking then historical drug usage. And we can see that there is a huge number of the significant associations, and a lot of those associations show similar effect we have seen in antibiotics. And again, when we do the associations with diseases and drugs, we can see quite a similar pattern, which telling us that there is a lot of confounding effect of different drugs. So again, when we study the diseases, it's very important to take account also the drug effect, to discriminate those effects between each other. So that's lead us to really design a new study. We were starting last week, and we do this in collaboration with Kelly Lehto, my good colleague in Estonian Biobank. And the idea behind was that, and in this first study, we saw this additive effect of the microbiome with antibiotics, but we had also a significant effect with the antidepressants. And we were thinking that we should study this in more closely, and we designed a study right now so that we had a baseline. Baseline means the microbiome cohort, which was collected in 2017-19, and now we started to collect follow-up samples, and we had defined 3 different groups. And the aim was to really distinguish depression-specific changes in the microbiome from these antidepressant-induced changes in the microbiome. And we have 3 groups. The first group is like a targeted groups. The people in a biobank within the 5 years, they have been treated with antidepressants, and they have also diagnosed a depression. And the second group is where we use the data which Kelly's group was collecting, this mental health online survey, which was done in 2021 in Estonian Biobank. And we have the very good overlap with Estonian microbiome data. Almost 70% individuals. And here we can define the group based on this depression screening tool, and they can define individuals who have depression diagnosed based on electronic health records, or who has depressive symptoms based on this mental health questionnaire. So basically those people haven't taken any antidepressants, and then we have a third group which is a control group. They don't have either disease and they don't take antidepressants. So we can really distinguish those different associations between microbiome affecting disease or effects of the antidepressants usage. So I'm very excited to have this data and to analyze this data, because our preliminary data, looking at the data which we have in the past and the past information from Estonian Biobank, we really think this is important, and thinking that antibiotics, doctors really know that it's changing a microbiome diversity a lot, but antidepressants, this is dark matter, and I think for doctors, because if we think about how many people are really nowadays taking antidepressants, and the young people, it may really influence the gut microbiome, which in turn may really change their health in the future. This is a final slide, which is kind of perspective, the view. What we really want to do in our research is that when we think about personal medicine, then we are interested to define people in a population who have a high risk in certain diseases, or we can improve the prediction of the diseases or diagnosis or do the better treatment. We have to consider also the microbiome, not only the human genome, and that's the landscape we do in Estonian Biobank, that we in the future may be able to do models where we count all those factors, of course the lifestyle, environment, the exposome, to define those high-risk individuals, to really be able to give some advice before. And there is just one study which is kind of leading this field. Is in Israel. They established 2 years ago already a very big cohort. This is 10,000 individuals. It's a prospective and longitudinal cohort where they follow up each year in 25 years to develop those prediction models, and they analyze all those individuals, all those factors which I mentioned here, and to be able to identify novel molecular markers to diagnostic, prognostic, and therapeutic values. And this is acknowledgement slide. This is my group, and my group was just the number of my people just increased recently, half a year before the story which I told you. We had only 3 PhD students, so it was a very small group. But I'm very happy that those very talented people were able to do huge work. Thank you.